Okay. Good afternoon, everybody. I'd like to welcome you to this webinar, which is part of the Innovation Nation series. And this particular one is on agricultural innovation. And it's a joint webinar between the Ag and Food Forum of ATSI and the Industry and Innovation Forum of ATSI. And it's my pleasure to welcome you all. Welcome for country. And as we gather for this meeting from different places around Australia and also overseas, I'd like to acknowledge the traditional custodians of the lands upon which we meet. I'm joining you today from Ngunnawal country. I pay respect to the elders, past and present, and also to any Aboriginal and Torres Strait Islander people who are joining us online today. And as we share our knowledge and our practices today, we also would like to acknowledge the deep knowledge forever embedded in the custodianship of the country. So welcome. So the housekeeping issues are, this is a webinar, it is being recorded and will be published on the Academy's YouTube channel. Now, if you have any questions during the seminar, we would ask you to put them in the Q&A function found in the Zoom ribbon. And we'll also encourage you to update any questions by liking them. If you want to have a chat in the side, that's also okay, but we would encourage you to put your questions mostly in the Q&A function because that's what Snow and I will actually be looking at. There'll be 3 speakers today and followed at the end by 20 minutes for questions and answers. So with that, I'd like to welcome you all and hand over to Snow Barlow, Professor Snow Barlow, who will actually introduce the webinar formally. Thank you, Snow.
Welcome to all participants from the Academy of Technological Sciences and Engineering Innovation Nation webinar series. As Australian agriculture has long depended on continued productivity increases to remain internationally competitive. And after nearly 2 centuries, we remain so, and we're a major exporter of food to the globe, and that can only continue as if we continue to innovate. So this series and the 3 speakers we've chosen today underline the innovations currently going on in Australian agriculture to address both productivity issues, but also to address environmental issues that are emerging around the use of chemicals in agricultural systems, around the emissions of greenhouse gases from our agricultural production systems, and in horticultural systems, the use of products that are essentially provide some protection against pest and disease that are not chemical, well, they're chemical, but they're not toxic chemical type products. So today we have 3 outstanding speakers that I hope you'll enjoy, and each will have 10 minutes. I'm going to introduce them all now so that when they finish their 10 minutes, they will automatically hand over to the next speaker. The first speaker, of course, will be Jidon and he is the co-founder and the CEO of Bilberry, which is a French startup company that seeks to use green camera technology to effectively, using AI technology, which he can explain if you wish, to target weeds in a crop so that rather than having to spray the whole area, he can target where the weeds are and therefore restrict chemical use and make everything more economic. The second speaker will be Eve Faulkner. Eve Faulkner is the Marketing and Communications Manager of Future Feeds. And Future Feeds is a CSIRO startup, which Eve will describe, to commercialize an exciting technology that has been developed by CSIRO and one university to limit the emissions of methane from ruminant livestock. And Eve will explain that. But it has dramatic results, and Future Feeds is seeking to commercialize this technology. And the third speaker will be Professor Nina Mitter. Nina is the director of the Centre for Horticultural Science at the University of Queensland Agricultural Science, QAFI, and also part of a CRC for innovation in this area. And she will describe her combination of BioClay and nucleic acid products to protect horticultural crops from pests and diseases. Nina, has just joined us. So we look forward to you speaking fifth. So without taking any of your time, I'd like now to ask Jourdain to begin by giving his presentation. Thank you.
Hello, so good morning. Good morning, everyone. My name is Guillaume Jourdain. I'm very happy to join you, and I would like to thank ATSI for the opportunity to speak. So I join you from France, and more exactly from the French Alps, where there's going to be a fantastic stage of the Tour de France today. But I'm not going to talk about the Tour de France. I'm going to talk about agriculture, and more exactly green-on-green spray technology. So I think the best is to start with a video of actually what we do. So what we do is we put cameras on spray booms. You can see the cameras here. One camera every 3 meters on the boom. The cameras in real time will scan the field. Then hopefully the video will work. Scans the field, then detect the weeds, make the difference between the weeds and the crops, and then we'll just automatically turn on the nozzles so that you just spray on the weeds and not on the whole field. So the advantage obviously is to save a lot of chemicals. I'll come back on that later. So just a bit of background about Bilberry. So I co-founded the company in 2016, and I actually moved to Australia in 2019 for a bit more than 2 years. So I'm now back in France, but I lived in WA in Perth for a while. We are now a bit more than 30 in the team, 7 people in Australia to focus on the Australian market. And so our main goal is to develop smart solutions for our growers. We do everything with technology, cameras, and AI, and we focus on what we call green on green and green on brown spot spraying. So how does it actually works? First, just a bit about the product. So there are different components that we develop and work with. The first one is hardware. We need some hardware, so the cameras, some computers, the display in the screen, lots of cables, all that we try to work with off-the-shelf component. Then we've got embedded software to make sure the cameras talk with the computers, to talk with the sprayers. So we develop all that in-house. And we've got the artificial intelligence algorithms to make the difference between the weeds and the crops that we also develop in-house. So how does it work? So the cameras are installed on the sprayer, as you can see here. So one camera every 3 meters. Then it's just a normal way for the growers. So they drive over the paddocks with the cameras. Then the cameras take pictures. So that's a real picture from a camera. Then the images are sent to the computers, and that's where the AI works. So the red overlay that you see is that actually what the algorithm sees. What's interesting is that you can detect one type of weeds, or you can make the difference between different types of weeds. Here in red, the red overlay is duckweeds. I don't know if you can see them well, but there are also some green overlays on the dandelion flowers. So it can be interesting for some growers to detect different types of weeds. Now it's just a video to show how it works in real time. I hope it's going to be quite fluid. So it's pictures actually taken by the cameras on the sprayer. That's why it's a bit choppy. It's 8 pixels per second. And on the in the white rectangle that you can see on top of the screen, it's actually where the cameras and the computers try and detect the weeds. Again, there is a red overlay when the algorithm detects something. Here, the application is to detect broadleaf weeds in cereals. So cereals here, it's wheat, can be barley or oat as well. And once weed is detected by the cameras, then it will send the trigger to open the corresponding nozzle to only spray on that specific weed, and then there's real-time spot spraying. So now just a question about why should we do spot spraying? So the first important thing about spot spraying is that since you can spray on the right spot at the right time, you can think differently about resistance management. Weed resistance is one of the biggest issues for growers, and they need new tools to be able to control those resistant weeds. And there's a very interesting question that we can ask with the cameras. What can you actually change in your spraying strategy if you can spray 10 times less herbicides? Because that's what we see on average in Australia, about 90% reduction in herbicides. You can also work on your seed bank management. From one year to the other, you can map the weeds. And so you have weed maps over the years of, I have this weed here, this weed there on this specific paddock. And knowing that year after year is a big help to know if your weeding strategy actually works. Cost savings is obviously a big one. You save 90% chemicals, meaning that your budget is just divided by 10, your chemical budget. And herbicide is the biggest part of the crop protection budget for Australian growers. You can also reduce crop damage, because even if herbicides are made not to harm the crop, there is still an impact on the crop. So not spraying the crop helps reduce that damage and hopefully increase yield. The last point is obviously around sustainability. Everyone wants to move towards a more sustainable world, and we need to reduce emissions and chemical usage in general. So that's a big tool to work in that direction. Now just a few words about where we stand. We can be found in lots of different sprayers in Australia. We work with more than 50% of the sprayer manufacturers. We've got different ones there, Gold Acres, Australian spray manufacturer. We also work with robots. I don't know if you've heard of Swarm Farm. That's the picture, top middle picture. So it's a completely autonomous robot with cameras on it who can drive without anyone there. So it's an interesting project that I can talk more about if you want. And I think that's it about the introduction of Bilberry. You can follow us. I think the best is to follow us on Twitter. We try to post a lot just about what we do in the field, and lots of customers, they also post their and share their stories over there. So you can either follow the company, Bilberry Agri, or myself. You can go on your website, or again, very happy to answer questions directly on my email or during the webinar just after. I'll be happy to take questions. Thanks a lot for your attention, and yeah, look forward to the discussions.
Thank you. We might move to Eve.
Thanks, Glenn. Hi, everyone. I am Eve. I'm with FutureFeed, and I'm excited to share a bit with you about our technology. So FutureFeed is targeted on livestock. So livestock produce methane. Ruminant livestock, they have an ability to break down grass and other fibrous materials, but that process results in two things: energy-dense nutrients, the volatile fatty acids, but also the byproduct of methane. And it's a challenge. So 11% of Australia's total emissions come from livestock, and 48% of our methane emissions. And so what we have is an industry, and when I talk about industry, it's the red meat, dairy, and wool industries that need to act. and they need to do that for a few reasons they identified, and that is maintaining market share, creating new value for the sector. There's market access, particularly with cross-carbon border tax adjustments, very relevant and only going to become more so. And then also consumer demand and the opportunity for niche markets. The other piece of the puzzle is that as other sectors of their economy decrease their emissions, the proportion on agriculture increases. So while it's not huge at the moment, it may potentially increase proportionally over time. The great news is that there is an incredible seaweed called Asparagopsis that has an overwhelming ability to reduce methane out of the digestive process for ruminant livestock. And how it all came about, there was a farmer in Canada who noticed that the cattle grazing on the seaweed near his on his farm they were dairy cattle, they were producing a bit more milk, having fewer cases of mastitis, and he wanted to know why. So he took the cattle to Dalhousie University, where our chief scientist, Dr. Rob Kinley, was working at the time, and to check that it was safe and see if he could feed it to his other cattle that weren't near the water.
Eve, sorry, could I interrupt? Yes. A number of us just have a blank screen instead of your screen. Is that right?
No, that is not right.
Okay, just keep talking and we'll see how we go. I'm sorry.
That's okay. I can try again.
There you go. Excellent.
We can now see.
Great. I'll quickly go back one so that you can see what Asparagopsis looks like. There we go. So this is Asparagopsis. So it came about, the farmer in Canada was, he took his cattle to Dalhousie University and they did some testing, and as part of that, our chief scientist, Rob, as all great scientists are, was curious as to what the methane reductions were doing. And what they noticed was about a 10-15% reduction in the methane being emitted from the cattle eating any seaweed that grazed. So that led Rob on a worldwide search where he joined up here with CSIRO, Meat and Livestock Australia, and James Cook University to look into the potential for seaweed to lower methane in ruminants. So they trialled over 30 types of seaweed, and the results on Asparagopsis were so good that they thought they had a testing error. They tested not once, not twice, but 3 times, and it wasn't until the 3rd time of the virtual elimination of methane that they really believed that they were onto something incredible. And so there are some kind of at a high level, it's found in nature. It's a seaweed that is native to here in Australia, and it's backed by a lot of science to say that it's safe and that it works. It only takes a little bit, so it's around 5 grams of Asparagopsis per kilo of dry matter intake to make over an 80% reduction. There are, and I'll have a look at one of these studies with you quickly, but studies have indicated that average daily weight gain and feed conversion efficiency can be improved at low inclusions. And there's also a long chain of benefits. So we're building an entire industry that's around the cultivation of the seaweed, as well as then the end impact of feeding it. So here is some of the results from, can you see my cursor? I'll work on the assumption. So here at 96 hours, you can see a virtual elimination of methane in the in vitro testing. And then there was a really exciting discovery. You know, sometimes things will work in vitro and you take them into animals and you don't see the same results. But actually what they found when they took them to animals was that they required even less seaweed than they did in vitro to have a huge impact. So there are lots of published studies, but I'm going to talk to one in the interest of time today. And that is one published in 2020 by Dr. Rob Kinley, our chief scientist, that looked at it at a 0.05, 0.1 and a 0.2% inclusion. It was a 90-day trial. And what it saw here, you can see over at the 0.2% inclusion, was a 98% reduction in methane. And that the cattle, you know, they were willing to eat the seaweed. And what it also showed, very excitingly, was improvements in weight gains. So in this one, we saw a 20% weight gain improvement in the 0.2% inclusion, with no changes in meat quality or taste. The numbers around the productivity are a little noisy, so we're doing a larger scale demonstration of that this year to get some more statistically relevant data, but the early indicators have been very positive. So then comes in future feed. So CSIRO has a program where when something a discovery like this, they will spin out a company, and with support from industry. So our investors are Woolworths Group, Grain Corp, Andrew Forrest Harvest Road, a group called AGP Sustainable Real Assets, and Spark Labs, and then CSIRO. And it's our job to commercialize the technology around the world. And we do this largely through license agreements. So we have the rights to license there are 2 patents that exist around the use of Asparagopsis. They're method patents. And we license impact partners around the world to take the seaweed and sell it into the livestock market with a methane reduction claim, and potentially in the future a productivity improvement claim. We've got 7 globally. Australia is leading the charge with 3 here in Australia, and we've got 2 in the US who operate out of largely Hawaii, then 1 in the EU and 1 in Canada. And what our licenses do, it's about quality assurance and trust in the industry. So it means that we can set things like minimum quality standards and requirements to ensure that, you know, when you're rolling out something new, that it's rolled out with rigor and with a due process. But what we recognized was that it takes more than licenses for a new industry to flourish. And so we focus on another 4 kind of key areas. The first being research. So significant investment in a research program to further the foundational science. And as I mentioned before, with a focus on productivity, next generation products, and also then grazing systems. So currently the science is applicable in a feedlot setting. And then there's an industry-wide push on at the moment to how we take supplement feeding for methane reduction out to an open grazing environment. There's certification. So we're building a global certified trademark that will travel all the way along the value chain to give consumers confidence, and also buyers at every stage confidence that the products being sold meet the requirements that our standards and our science commands. We're opening regulatory pathways. So that looks different in different regions, but working on our scientific dossiers that pull together the information. And then the bit that I get to do a lot of, and that is we get to tell people about it, and we do a lot of sharing of the technology, and then that will go into our certified trademark in the future as well. So what we're focusing on at the moment is impact and scale. So there are a few things the industry needs to succeed in that sense, and prioritization. So looking at carbon accounting methodologies, some regulatory updates. So that differs in different markets, but getting to a point where both current and next generation products can be fed in all markets. It is a really collaborative industry, and there's I think collaboration in new technology is such an important piece of the puzzle. And then investment. So there's been about 100 million invested in the Asparagopsis industry, not in FutureFeed, but in the broader industry over the last couple of years. And so lots coming up in the future. And that's my summary. Thank you. I'll pass over to Nina.
Nina, can you begin now?
Thank you so much. No, thank you. Thanks for this opportunity. And yes, I'll start sharing my screen, and I hope it all goes okay.
Yes, we can see it.
All right, fantastic. Thank you very okay. Thanks again. And before I start, I must begin by acknowledging the traditional custodians of the land from which I am talking, the Turrbal and the Yagara people, and pay my respects to elders past, present, and emerging. You know, we have all, I think, become very familiar with the powerful world of RNA, and it's not a pleasure to say that, because we have become familiar because of now we will be getting the 4th dose as well. And we know that there are those RNA vaccines, which know Moderna and Pfizer have made us kind of look into. But what I'm trying to talk today is the world of RNA for agriculture, or for managing pests and diseases. And this is the work we started about 10 years back, so much before these RNA vaccines came around. I think agriculture, I always feel, has is the genesis of many new things that come from the plant side of things. I'm really biased about plants. So today what I'm going to talk to you is RNA-based biopesticides as a sustainable way of crop protection, and especially the technology called BioClay that we have been developing and is progressing quite well. If I look at pesticides, I do not think anyone needs to be reminded of the issues of resistance development. Pests and pathogens are very clever. You develop a pesticide product and they can develop resistance to it. There is the issue of residue on our fresh produce, health and environmental issues, runoff issues to our precious waterways. There's also that lack of specificity. We do not want to harm our beneficials when we spray these pesticides, and new chemicals are hard to come by. However, I'm not saying that we can live without plant protection or pesticides, because even with the use of pesticides, we still lose 20 to 40% of our production. So we do need them, but how we can approach a bit of a reduction scenario in them, or have some alternatives that can work as an integrated pest management tool, could be the vision moving forward, especially with world now, setting targets. I was in Europe last week, and there they have set a target of 50% pesticide reduction by 2030. So these things are going to have an impact on our economy and export and trade as well. For me, the trigger, I cannot just get rid of this slide from any of my talks, because this was the trigger for me in 2013, actually, to work on this area of RNA-based biopesticides. When I read this, pesticide contaminated community meal kills 25 Indian children. So it still is happening, and therefore looked at the things that we were doing and decided, okay, let's see if we can find some alternative to pesticides. And this is where the genesis of RNA-based biopesticides or BioClay happened. The word bio in BioClay is actually RNA as the biological active. So instead of a chemical active, which is present in your fungicides or pesticides, in this case, ribonucleic acid or RNA is the biological active, and this RNA actually comes from the pest or pathogen itself. And clay particles are just the carriers for this RNA, and hence the word BioClay. RNA on its own, when you spray on a crop, is very fragile. It won't last for more than 3 days or 5 days in the field situation, and it'll get washed off by rain. Clay provides that carrier which can provide it stability, and where it does not get washed off. So there won't be any residue. And this clay is completely biodegradable. So we hit on those buttons of being no residue, specific, stable, sustainable, safe. And most importantly, again, from a consumer acceptance perspective, this is non-GM. We are just spraying the plant. So what is BioClay? It is just an inert biodegradable clay. It is nothing but magnesium and iron as a clay particle. And we deliver the RNA using this clay. It provides clay layers degrade naturally, leaving no residue, and we get extended stability. As I said, this RNA comes from the pest or pathogen itself to kill the pathogen. So for me, it is almost like nature versus nature with this particular tool. And yes, it does work. It's taking us time. It has got its ups and downs, and we have lived through many failures and then successes and then lived again. It really does work. And this was the work we published in 2017, actually. And I'll take you through the progress of this work. But just to show, if you look at panel A, BioClay, those black blobs are the RNA that we spray. So BioClay can survive for more than 30 days on the plants. And if you look at panel D, it can just provide the leaves. Sprayed leaves are protected from virus infection even after 20 days of spray. So we are extending that window of protection. I must mention here that this is not a lifetime. This RNA technology is just like any other fungicide or pesticide spray. You'll have to spray again depending on the crop cycle, season, when the pest comes. The protection window we are aiming for is about 8 to 12 weeks. It does work. So since then, we have moved on and showed that the system works for multiple virus diseases. This is tomato spotted wilt virus on capsicum plants. And as you can see here, the control plant is looking almost dead due to virus infection, whereas a BioClay sprayed plants does survive, and we have collected yield data and plant data, and this looks like a really healthy crop. We had also worked with zucchini yellow mosaic virus. Now, this is again a major virus of these zucchini crop, and we have done this in a field trial as well. As you can see here, what is the difference between a plant which has been just sprayed with water versus the plant which has been sprayed with BioClay and then challenged with the virus. We are still optimizing the field application system. We want to make it much more efficient, but however, the results are very, very promising. We started working with viruses and then moved to insect pests as well, realizing the economic significance of this pest. So we just published actually last month another paper in Nature Plants, which is about clay-delivered RNA interference for whiteflies. Now, whiteflies have a very huge host range. They infect more than 800 crops, cotton, broadacre, horticultural, not only as a pest, but they transmit a large number of viruses as well. So they are really damaging to the crops. And in this case, we focused on cotton and tomato. And I'm very pleased to say even ABC Landline covered it this month along with the cotton growers. And this work was funded by Cotton Industry, Hort Innovation, Queensland Government, and New Farm Australia Limited as our commercial partner. This is just to show that this works for insect pests. You spray the crop with your RNA. The RNA will go into the plant, will remain in the plant. When the whitefly comes and feeds on such a plant, this orange thing that you are seeing inside the whitefly is the RNA which it has sucked from the plant. And this is the RNA that we sprayed on the plant, and whitefly has sucked it. And now this RNA is going to kill the whitefly because that is what it is meant to do. And we not only showed for adult whiteflies, we have even showed it for that this works for even nymphs or eggs when they are laid on the surface of the plants. And as you can see here, these phenotypic effects of RNA BioClay on whiteflies, whether it is an egg or a nymph, they do not develop. We get death of whiteflies as well. Most importantly, in this work, we also showed that this does not have any effect on stingless bees. So we not only designed the constructs in a bioinformatics way that we were saying it won't have any effect on any other species other than whitefly, but we actually physically did the experiments and showed as well that feeding bees on this RNA does not harm them in any way. So this was once again another important step. We have now moved to fungal diseases as well. So we are working on insects, pests, viruses, but fungi became the focus because of the magnitude of damage that fungal diseases cause to both broadacre and horticultural crops. We have got now an Australian Research Council research hub, huge hub having, so very pleased to say the team of two that team of three that we started with in 2012, 2013 has now grown to a team of 50, all working on this BioClay concept with New Farm Australia as the commercialization partner, multiple universities, almost all RDCs, state governments and industry partners have come on board with this hub. This slide is from our international partner in USA. Here, the green color that you are seeing is actually the RNA. So we have labeled the RNA. This is just to show as fungi, when they infect a plant, they take up the chemical ingredient when you spray a chemical pesticides. In a similar fashion, they can just take up the RNA as well. So the RNA from that sprayed plant can actually enter into the fungus. And in this case, then this RNA will kill that fungus. This is just another example of it working on chickpea crop. So you can see the difference between water, naked RNA and BioClay to show that BioClay does provide an extended protection window, and we are optimizing these experiments further. Very exciting work one of the postdoc and the PhD student is doing in our lab, and Sawyer and Beck, where they are looking at myrtle rust along with DAF Queensland. More than 300 Australian myrtaceae species get infected with this rust and almost threatens whole ecosystems, and very promising early results, as you can see, between control plants and double-stranded RNA in managing rust infection. Now, this can have a bit broader implications if we start looking at other rusts as well. The research hub process, just to briefly, that our philosophy is not just research, but with a vision that it needs to have an end vision and a product development. So the hub process is designed in a way that it goes from synthesizing lab glass house all the way to field with scale up, but also having some very important elements of registration, regulatory framework development. And most importantly for me, I think, is the social licensing aspect of it. So we have built in that component to start straight away with communication strategies, adoption strategies. How do we reach and how do we communicate this concept for wider adoption? So those are some of the key features that we have kept in mind. As I mentioned, I was saying it's non-GM right from the start, but it took me some time. Now the Office of the Gene Technology Regulator in 2019 has also passed the legislation that topically applied dsRNA is exempt from any GMO regulation. So it does not need deregulation, and it's the same case in US, Canada, Japan, Brazil. Europe is still undecided, but let's see where it goes. Another advantage of this technology in terms of trade and markets is because it does not have any residue, there's no maximum residue limit. So we are looking at some advantages here. And of course, when we look at this technology, we look at in a holistic way of its role in the food and agribusiness value chain, not just about, it is, about that research that will develop that, but having a lens of sustainability, having a lens of enhanced production and value, looking at the global marketplace, as well as looking at the future consumer who wants to know the provenance. So just in the end, wanted to say, innovations really with this BioClay platform, still a way to go. We don't have a product on market. We are working with New Farm Australia, trying to resolve those issues that are still needed to take that development further. And endless possibilities, whether it is viruses or insects or in controlled environment agriculture or in pack houses. One of the postdoc, Dr. Karishma Modi in the lab, is actually looking at animal using the same philosophy for animal health targets as well. So there is that branching out, but design of regulation and public opinion are crucial, and we need to keep that in mind. As Australian owned and invented, so feel proud of that it is important innovation. Hopefully it would make some difference to the health of our planet, and couldn't have done it without these contributions from these wonderful partners who have come on board. So a truly collaborative approach here. Thank you.
Thank you, Nina. Joanne, you're there. Do you want to ask the first question?
Yes, I can actually. I was going to go back to Guillaume and ask, we have a question from an anonymous attendee is there any loss of weedicide due to wind drift?
Yes, so that's a very good question. So I think wind drift is a huge issue in agriculture because yes, there is a lot of loss of herbicide, but also because there can be contamination of other fields. For instance, if you've got a canola field next to a wheat field and you are spraying your herbicide on your wheat, the herbicide will not damage the wheat, but could damage the canola. And one of the biggest risks of drift is that actually, if there's a lot of wind, the chemical will just drift onto the canola field, and that can lead to just dead canola and loss of yield or loss of crop. So that's a problem in general, I would say. And this problem of drift is reduced with smart spraying because you spray less, so you're less likely to have a drift going onto another field. That being said, it's also a challenge because you are not supposed to spray if it's too windy, but still Australian farms are so big that if there's a bit of wind, growers will still spray because of the operational constraints. And so when there's wind, we've got to adapt at the moment at which we open the nozzle because we have to take wind into account, otherwise the spray will not get to the weed. So usually the way we do it is that we just depending on the conditions, we add what we call overspray, so a bit of margin before and a bit of margin after the spray, so that we make sure we compensate for the wind, but also for other elements such as, for instance, the movements of the boom, because the boom can move up and down or just back and forth. And so we are just compensating for that thanks to overspray.
Okay, thank you.
Thank you. A question for you, Eve. This question is from Neil Turner in the Western Australia. He asks, the animal rumen, the ruminant rumen, is incredibly complex microbiological soup. Do you have any indication as yet about whether the microbes in that rumen adapt to seaweed, and so the effectiveness of seaweed may decrease with time if other bugs emerge that are able to not be affected by the seaweed product?
We have looked into that. We've got our longest study, or the longest study done in the space to date, is 147 days, and that showed no adaptation across those 147 days. So it showed that it continued to be effective, which was great news, really good news. There is a 300-day trial happening at the moment, but certainly none of the indicators would suggest that the rumen's adapting at this point, which is great.
Thank you. That's very helpful. And Joanne, do you have a question for?
Well, I have a follow-up question for Eve, and then I'll have also a question for Nina, but I'll ask that after you. So Eve, is this mostly going to be targeted at feedlots rather than sort of more grass-fed environments?
So initially, yes, and that is when we've taken, I guess, a staged approach to the science. The feedlot's the easiest space for us because you can control what is going in with every mouthful. You can mix the seaweed through all the feed. What we do have underway, both internally and then the federal government has a program, a grant program to support this, is how do you take it to a grazing system. So we've done some trials in pulse feeding where you feed twice a day. That's had really good results, and then gradually kind of stepping that out. The challenge with grazing is twofold. It's not just how do you deliver it, which is one piece, it's probably the easier piece of the puzzle, but how do you accurately then account for the methane reductions that might happen in a big herd across, and that's important when you come to things like carbon credit creation and accounting. So we're confident, given the foundational science and the robustness of it, that there will be a solution out into grazing, and there are a lot of people coming together at the moment to work on exactly that. For seaweed and really all methane reduction supplements have that same challenge in terms of how do you get it out there and how do you account for it.
Okay, thank you. Snow.
Nina, that's some very exciting work your group does, and it's quite a group now. What do you envisage the future here? Do you envisage in horticultural crops, with you and I both know very well that there are, multitudinous of pests and diseases that like to chew on them. Do you envisage that there might be a total biopesticide control of pests on those crops, so that effectively you will be applying no chemicals, but just bios?
Oh, good questions, No. And for me, I feel that the future is about integrated pest management. I don't think so we can have just any one solution as the silver bullet, as horticultural crops are complex, as you have said, not only the crops, but the pest and pathogens, such a wide host range of pests and pathogens as well. So I think what we need to be very realistic about is we will still need all those cultural management practices as well, all those other tools and techniques that we have in our arsenal as well. Yes, biological control, and not just BioClay, but other biological controls as well, which are emerging, would be an important factor. Pesticide companies may also pick up, I think improved chemistries will also play a role. So it would still stay for some time to come, at least that would be the case till something else changes.
Yes, I strongly agree that I think it's heading in the right direction. Joanne, do you have a question?
Yeah, I do. I have one for William, and it's from Beth Woods. She apologises if she missed this in your talk. Do you have algorithms for a range of crops and weed combinations, or is the technology currently limited to a small set of crops?
Yep. So great question. So our focus, first, our main focus is on Australia. And so we started with wheat, as it's just the biggest crop. And everything that we do, we keep at the center of everything the grower. And so we looked at the grower crop rotation to see what are the other crops that they are working on. So Australia, basically, when usually it's about half to 2/3 wheat and barley. Then you've got canola as your main rotation crop, and then usually you've got a legume bread crop, mostly lupin in WA, and then chickpeas and lentils over east. And so this year, for the very first time, grower will use the cameras on the whole crop rotation. So first, they spot sprayed some for their weeds for summer spraying, so in between crops, just what we call green on brown, so spraying all weeds on a bare soil. Then they are using our broadleaf in cereals algorithm to target broadleaf weeds in cereals. We are also detecting grass weeds in canola and different types of weeds. Actually, we've got 3 different algorithms for lupin. So we can detect blue lupin in lupin, which is quite interesting because that's a huge topic in WA. The lupin hectares are just decreasing because of blue lupin, because there's no solution, and cameras could actually maybe change the game. So that's a very interesting one. So yeah, we are developing an increasing number of applications, but that's still a work in progress.
Eve, a question for you. We noticed the current state of research you're concentrating on the feedlot industry. Have you done anything, or are you aiming to, the other livestock industry that actually does have a lot of containment and see the animals twice a day is clearly the dairy industry. Are you doing anything in the dairy industries, or do you have plans for that industry?
Yeah, definitely. So that twice daily study that I mentioned before was in dairy cattle. And exactly that, because you've got them, it's a great stepping stone because they're coming in twice a day to be milked. And so you have the opportunity to give them the supplement at that time. So big focus on both beef and dairy. And there's some work happening in wool as well.
Yeah.
Great.
Thank you. Joe?
Yeah, look, actually, it's very exciting. These 3 technologies are quite wonderful, aren't they? So, Nina, we have a question here, which is to do with the cost, the relative cost of producing the mRNA and its synthesis compared with conventional chemicals. Do you have a sense of that at this stage?
Yeah. Thanks again, a good question. And yeah, what we use is double-stranded RNA, so a little bit different than the messenger RNA, which is used in vaccine production, but cost is a critical factor. The aim has always been that it should be either similar to the pesticides cost or a bit lower. When I started the work, it really looked unachievable, Joanne. To be honest, it was really my most fearsome thing, that what about cost? Because we were buying at that time RNA for maybe $2,000 per gram. So even research was difficult to do with that kind of a cost, leave aside the practical things. But the technology has moved so much that now there are companies are quoting us $2 per gram. So you can imagine the difference that has happened in the space of double-stranded RNA production, especially for agriculture, because we do not need that GMP kind of certification, which is needed from a human health perspective in terms of mRNA, and therefore that cost has come down and technologies have evolved. And clay is very much in the economic range to produce, so that's the new farm target as well. So hopefully we will be in good space with the cost.
Thank you.
Thank you. A question for you, Gianni. I know that you have an office in Western Australia now. But you did say your company, Bilberry, is operating in 7 countries. So what are the other countries where your company is operating?
Yep. So actually, we've got other product lines. So for the product line that I presented today, we do 95% of our sales in Australia, and we have just opened Canada this year. We are working actually in more than 10 countries all across Europe for rail spraying. So we do exactly the same. We put cameras to detect, or we use cameras to detect weeds, but not on agricultural sprayers, but on trains and actually trucks and quads and everything that would go on rail tracks, because there's a need. It's for safety reasons that you need to kill the weeds, because there can be, if there are too many weeds, there could be water infiltration into the soil, and that can lead to some movement of the actual trucks, which can lead to big incidents. So that's why you want to control the weeds, and so we just put cameras. And in Europe there's a huge pressure around sustainability, and now in most of the tenders, because it's public tenders, the people that are doing the weeding, they have to show that they are using techniques to reduce the amount of herbicide that they use. So cameras is almost a must now in Europe. So that's why we can work in different countries on that product line.
Yes, thank you. Joanne?
Yeah, I've got another question for Eve. It's actually two questions, and I think I'll combine them. So it's really about the cost of production and availability. So the cost of growing, harvesting, drying, transporting, and treating the seaweed, and its availability may be limiting in its role as a supplement. Can you comment on that aspect of its cost?
So the market will determine cost. Future Feed doesn't set a cost. It is up to licensees in the market. What we do anticipate will happen is that, as you see with most new technologies, costs will come down over time as production becomes more efficient. There's also the piece around the productivity improvements, potential productivity improvements that could help to offset the cost of the supplement. And then as carbon prices increase, there may be a value piece there, but also as taxes potentially increase, as we're seeing in places like New Zealand, the kind of overall cost benefit will be determined as the market progresses. We're still, we had our first commercial sales here in Australia last month, so it is still a very nascent industry, and I imagine it will determine it over the next little bit.
Yeah, there was also a question from Beth about, it's rather critical about the options for delivery to grass-fed beef, and I'm wondering if you want to comment on that, but in particular, what proportion of methane emissions in Australia come from feedlots versus grass-fed beef? Do you know that?
It's a great question that I don't have off the top of my head.
Make it up. No. Can you guess? Do you have an rough idea?
I think a good start is just considering the number of cattle that are in feedlots as opposed to any one time, and that number of cattle are free range. So at this point, the majority still would be free range, I think.
Yeah,
they often come into feedlots as part of their life cycle, but I don't know. But I would say more than 90% probably. But
I think it is
somewhere.
And that's exactly it. We kind of don't let perfection get in the way of progress. And that is, if we can really successfully target those feedlot cattle today and then move towards the others as supply increases as well, there's a nice timing synergy there.
So one of the comments is that about 50% of cattle are currently feedlot to be finished off before being sent to market. Yeah. So,
Nina, clearly you regard this BioClay, RNA BioClay, as a platform. And we both know that, and I noticed, I was fascinated by your work with whitefly on cotton. Well, I guess the question is this: a traditional problem with horticultural crops has been the small acreages, and how do you spend the money to develop biopesticides for what might be individually smaller crops, but in aggregate they add up to a very big sector. So how are you balancing the concentration between the horticultural concentrations and those much more broad acre concentrations with things like cotton?
Once again, very good questions, no? And so what we are at the moment, as Evans mentioned, progressing on certain critical things. So in the suite of things, we do look at, as a start, we are looking at pathogens which really, in fact, have a broad host range. So, the fusariums of the world, or the, white flies, as I talked about, and looking at something, which, even for viruses, we are looking at viruses which have a very wide host range. They do not affect just tomatoes or just chilies. They have maybe a host range of all Cucurbitaceae family or wider. And that would give us, in the terms of production and cost targets, which can work across. Similarly, for pathogens, we are looking at all global diversity of isolates, so not just focusing on, this is the whitefly population in genomics in Australia. We are looking at the global populations of those pathogens and the genomic data in our design of the BioClay. So as it evolves, those will be the steps, which will then provide us with answers, working on broader pests and, of economic significance first, and then looking at how do we then tailor it to, as you are very right, to some of the niche but small horticultural crops as well.
We're just about out of time, but can I ask you a sort of supplementary on that? Is because it is a biological, does that in any way circumvent the residue limits that operate in not only across national borders, which differ, but also in Australia and in state borders? So do you envisage that it might be easier to, if you like, cross-accredit these biopesticides across, well, certainly state boundaries, but hopefully national boundaries, because it's not about residue limits, it's just about agreement on the gene regulation, essentially.
Yes, exactly. So if I'm not, to be honest, with this particular technology, residue does not become an issue for me. It is the dead deregulation, and I'm very pleased, at least we have a deregulation now in Australia. So the GM regulation is not required. We just have to register the product with APVMA as a biological control product, and those registration packages will be still required, and whatever be the, their dossier requires us to do. And this would be also then not only nationally, but internationally, wherever this topical RNAi does not require GM regulation. In terms of residue, I know now countries are setting up either nil residue limit or 2 ppm or, really low, but I believe that with BioClay, we will not have any issues with the residue limits. So
this webinar obviously could go on forever. 3 different but absolutely fascinating new technologies, all which are very, very promising. So can I briefly thank the speakers, but ask Joanne to close the webinar? Thank you.
Yeah, I'd also like to add to Snow's comments, because these 3 challenges these speakers have spoken of have been very significant issues for agriculture for a long time, with no clear path to solving them, I think, until these sorts of innovations are coming along. So I also want to thank the speakers, and thanks also for the participants for the questions. And so with that, I'd like to draw this very exciting webinar to a close. Thank you.
Thank you all, and we'll draw it to a close. Goodbye.
Thanks.
Thank you, Snow.
Thank you.
Thanks, Joanne.
Thank you.